Okay, carbon dioxide reduction. Remember the definition of reduction. Reduction means the gaining of electron or hydrogen atom. So in this case, the definition is the gaining of hydrogen atom. The gaining of hydrogen atom, because you want to make carbohydrate, okay, from carbon dioxide. Carbon dioxide, we only have carbon and oxygen. We don't have hydrogen in there, so we need to give hydrogen to the carbon dioxide, and in this case it is called reduction, so that the carbon dioxide will become carbohydrate. So remember, at the end of carbon dioxide fixation, we make 3Pg. We make 3Pg molecule, and then 3Pg molecule undergo reduction to become G3P in 2 steps. Okay, G3P again, I tell you, it is like the precursor of glucose. And then we need to use ATP and NADPH from light reaction. Let's take a look. So 3Pg. Again, 3Pg is a 3-carbon molecule, 1 carbon, 2 carbon, 3 carbon. We get this 3Pg from the carbon dioxide fixation, and then we use this ATP, so ATP becomes ADP. By doing this, then one of the phosphate group from ATP will connect to the G3P. It will connect to the 3PG. From 3Pg, it becomes 1,3-bisphosphoglycerol. 1,3 tells you the 3. Okay, let me explain to you the name. 3 tells you that it is carbon number 3. So we have carbon number 1, 2, 3. It is the carbon number 3 that has the phosphate group, phosphor. That means this one here. The phosphate group is attached to carbon number 3. Now, using the energy coming from ATP, and by removing one of the inorganic phosphate group from ATP, it becomes 1,3-bisphosphor. What does it mean? So, again, 1, 2, 3, carbon number 3. Carbon number 3 still has the original phosphate group, just like the 3Pg at the beginning, okay? But carbon number 1 now also get a phosphate group from ATP. That's how we call it 1,3. 1,3 tells you which carbon has the phosphate group, phosphate group. But why do we call bis? Bis meaning that, oh, you have 2 phosphate group. Bis like mean 2. It's like bi. Bi means 2 phosphate group. So the 2 phosphate group is connected to carbon number 1 and carbon number 3. And then you have an enzyme called glyceraldehyde 3-phosphate dehydrogenase. Okay, this is a very long name. Glyceraldehyde G3P. Basically, glyceraldehyde 3-phosphate is the G3P, and you just put dehydrogenase. Dehydrogenase. Let's take a look what's going on. So again, you have 3 carbon, 1, 2, 3, okay. But what happened in the carbon number 3 is that the phosphate group becomes the phosphate group becomes a hydrogen. Yes, the phosphate group becomes a hydrogen atom. Yes, the phosphate group becomes a hydrogen atom. So where is the hydrogen atom coming from? The hydrogen atom comes from the NADPH. It is the same hydrogen. So the enzyme G3P dehydrogenase. What's the function? It dehydrogen. That means D means remove hydrogen. It removes hydrogen from NADPH and then add this hydrogen atom into this gigantic molecule, so that the 1,3-bisphosphoglycerate will become G3P. So that's the purpose, the name of this enzyme, the function of this enzyme, is to remove the hydrogen atom from NADPH and put this hydrogen atom onto the 1,3-bisphosphoglycerate. You see that the phosphate group is removed here. The phosphate group is removed here. So this phosphate group is the same phosphate group as is this phosphate group. They're the same phosphate group. Okay. Removing this phosphate group and then add the hydrogen atom from NADPH onto the carbon number 1. Then we will have glyceraldehyde 3-phosphate. Again, the number 3 here tells you that the phosphate group is attached to carbon number 3 here. This is carbon number 2. This is carbon number 1. Okay? So that's a G3P is called glyceraldehyde 3-phosphate. Oh, sorry, I forgot to mention this. The enzyme for the first step is called phosphoglycerate kinase. The word kinase, this enzyme kinase always do phosphorylation. Phosphorylation. There's an R here. Phosphorylation. Meaning that kinase always put a phosphate group to another molecule. Kinase always put a phosphate group to a molecule. We call it phosphorylation. So you see that phosphoglycerate basically is the name of this molecule. They are the same one, phosphoglycerate. Meaning that, okay, this enzyme, phosphoglycerate kinase, what's the function? Kinase put a phosphate group to where? To the molecule phosphoglycerate. So that's basically the meaning of this enzyme. The name of the enzyme basically tells you the function. Kinase put a phosphate group to a molecule. Which molecule? Oh, phosphoglycerate. So this enzyme put a phosphate group in the carbon number 1 of the phosphoglycerate, then it becomes 1,3-bisphosphoglycerate. Then what is the function of the enzyme glyceraldehyde 3-phosphate dehydrogenase? Well, dehydrogenase meaning that it removes the hydrogen atom from NADPH and then put this hydrogen atom and put this hydrogen atom into carbon number 1, and then it becomes a G3P. So dehydrogenase make G3P by removing the phosphate group and adding a hydrogen atom. Okay, reduction of carbon dioxide. This is like the summary, okay? The previous slides, okay, you don't need to know all the structure that's so complicated. Just, if you really want to memorize it, just memorize this picture. If it's good enough for me, good enough for you. And you have a 3PG, the product of the from the carbon fixation. And then you used ATP. ATP become ADP. So you have this inorganic phosphate. This inorganic phosphate is added here, 1,3-bisphosphoglycerate. And then you have this NADPH. And the hydrogen atom here is added is added, and then to here, then you get G3P. So as 3PG becomes G3P, ATP becomes, okay, this is, and you have to put something here. The phosphate group is, the phosphate group leaves. The phosphate group goes away from BPG, so that the hydrogen atom from the NADPH can bind to the first carbon in G3P. So yeah, that's all you need to know for the reduction of carbon dioxide. Is the conversion of 3PG into G3P, okay, and using ATP and NADPH. Okay, there's no change in the number of carbon, meaning that during the carbon dioxide reduction, remember it starts with 3PG. It starts with 3PG, 3PG. 3PG. 3PG has 3 carbon. And then carbon dioxide reduction, it ends with what? G3P also has 3 carbon. So it's always 3 carbon molecules, okay? Hydrogen is added to the 3 carbon molecules, then you got the carbohydrate. So if you have 6 carbon dioxide, which is the overall equation of the photosynthesis we want you to memorize, then what will happen? Oh, this is wrong. It is not 12, it should be 18 ATP. So 18 ATP and 12 NADPH are spent during this stage. Okay, this is the last stage of Calvin cycle. Congratulations. Last stage of Calvin is the regeneration of RUBP. We call it a cycle because RUBP, at the end of it, it will show up again. We reuse the RUBP. So at first we use RUBP in the carbon dioxide fixation. How do we regenerate RUBP? Remember from the last stage, it is called carbon dioxide reduction. We make G3P. In other words, how do we generate RUBP from G3P? How do we generate RUBP? 3. So it said that every 3 turns. Okay, 3 is the key words. Every 3 turns of Calvin cycle, 3 is the magic number that you need to memorize. 3. It will generate 5 G3P. Remember, each G3P is a 3 carbon molecule. To make 3 RUBP, you need 5. RUBP is a 5 carbon molecule. So what does it mean? Okay. What does it mean? 5 times 3 is still 3 times 5 here. It means that, remember counting the carbon number. Okay. You have 5 free carbon molecule. So 5 free carbon molecule. And then you have 5 carbon molecule. That's the whole point. Okay, that's what it means. So every 3 turns of Calvin cycle, you can generate 5 G3P, and and also at the end of this, you can generate 3 RUBP from the 5 G3P molecule. From the 5 G3P molecule, you can generate 3 RUBP at the end of the Calvin cycle. Okay, let's take a look